And there's my art. There's already my first audio mistake. I thought it switched when I switched to the face cam, but I guess not. So I'll say hello again. Welcome on into the stream, everybody. Keep it making small adjustments here. All right. That should work. That should work enough. All right, I think we're going to go ahead and get started. Welcome on in, everybody. My name is Akamash, founder of Reef Unity Network. What we're going to be doing today is we're going to be taking a whole list of coral research data, so data measurements you might be collecting throughout your experiments or your field work, and we're going to be ranking them in terms of like how useful or how important I think they are to collect, and any annoyances I might have with them, or any other context or commentary I have on how these different measurements, these different metrics are collected, applied, any of the headaches I've had, any of the successes I've had, things I wish I had done or collected during my research. Hopefully you guys can take something away from this. You're like, oh, maybe I should be collecting that data measurement for my project. I hadn't considered that before. Or you might be on the fence with one and you go the other direction and you're like, maybe I don't need it as much, or maybe that'll be too much of a headache. This isn't supposed to be like, publication quality level analysis here as I go through this tier list. This is something that's just kind of fun, and I know I'm missing measurements, so if anyone here has ones that I missed that they want me to rank, put them in the chat. I'll be happy to add them here, because I can do that. So I'm gonna go ahead and pull this up. All right, here is our tier list. How this is going to work today. On the left, we have our different tier ranks, starting with S tier, basically meaning every project you conduct, any research you're doing, these are the metrics you should be taking. These are the measurements you should be collecting. Always. Then we have each one. A would be like, it's a good idea to have. You should always include it unless budget, time, constraints. So something you should probably always be doing, but if push comes to shove, something gets in the way, maybe you can get away without having it. B tier is like, yeah, good idea. You should be doing this. C, take it or leave it. Be nice to have, but not something I'd worry about if you don't need to collect it. D and F tier. So D tier. Probably means I've collected it in the past and I did nothing with it, or haven't done anything with it yet. So basically, I collected it. It didn't help me as much. I might consider not doing it in the future. And then F tier is like, I wouldn't bother with this at all, honestly. It's going to be similar. I may have collected it. I may not have collected it in the past. And I probably wouldn't do it for any of my future projects. There'll be some mix and match. There's probably too many here. I could probably cut one of them out, but not going to worry too much about it. On the right side, we have a bunch of different metrics. A lot of the ones that I kind of pulled from my projects, or I typically see people collecting in research papers. Again, this is not an exhaustive list. We'd be here all day. I don't plan on doing this all day, probably till about 3 o'clock. So we'll get through these. But if you do have recommendations on ones to add, throw them in the chat. I'll definitely hit them. I'll put them in and we'll rank them there. As I go through ranking these different ones, how am I going to do this objectively? Basically, I'm not. I'm going to go by what was useful for me, what wasn't useful for me, what was easy to collect, what was not easy to collect. I may move them across these different tier ranks as I go. Because, well, this is my stream. I'm going to do that. And none of these metrics are ever collected in a vacuum. They always, they're going to have some interconnectedness. They're going to have some relationship, especially if you're building models, complex statistical models with these, like they're going to interact a little differently. So if I put one in, say I rank it as an F tier, and then I decide, ooh, but this changes behavior when you include it with this other metric, I might change my mind and bump it up a notch. Do you think it's controversial? Tell me why. Tell me why, and we'll break it down. Well, let's go ahead. Let's get started. Start with this first one, genotype. So this could refer to either the coral host genotype or the symbiont, the algal symbiont, Symbiodiniaceae genotype. I have used this metric in two of my dissertation chapters. Both of them were, one of them was specifically on genotype interaction, so that was like the main metric, or one of the main things I was paying attention to, collecting, analyzing. So I'm gonna kind of ignore it from that study, because some more context on why I'm doing this is these should all be metrics that you collect in addition to your main variable of interest. So one thing you won't see on this list is like physiology. So like chlorophyll content or host protein content, because those are most likely going to be variables you are testing. Probably not something you're going to keep in the background. So back to genotype, one of my chapters, genotype, was actually kind of like a let's keep track of this and we'll see if it has any connection. So because of that, I'm going to give it an A tier. Obviously, we're going to get a lot of the it depends scenarios during this stream. But okay, let me form my thoughts here. Not all coral research is done with the host and symbiont. Sometimes you are looking at grazers, sometimes you're looking at holistic reef metrics, like benthic cover. We probably don't need genotype for that. So I'm putting it in A tier for now. If your study is using coral hosts or symbionts, probably want to know the genotypes. So I'm not putting in S tier because it's not always used. Next up, we have tank. So this is like the aquarium tank. If you're running an ex situ experiment, something if you have multiple tanks, you have multiple sources of variation. Some of the filtration might be a little bit different. Other random things might happen. This one I'm going to put also in A tier. Might change my mind on this one later. I had to include tank in the same study that I used for coral genotype because I was doing an ex situ bleaching experiment. We had multiple raceway tanks, and because of that, we wanted to make sure that each individual tank wasn't having its own effects on our corals' physiology or bleaching response. So it was really helpful to have. Thankfully, we didn't see any tank effects, but sometimes things can happen. A heater accidentally breaks and goes wild and spikes the temperature in your tank. That could create a tank effect. Or somehow the water quality degrades in one. That's a tank effect, and you can see that impact your physiology. So in that case, I'm gonna keep it A for now. I might change my mind. It might go down to B. We'll put a pin in that one for now. All right, sampling date. So this one would refer to when you collected your samples, whether it be you're out in the field collecting like sediment samples, which I have done, or even just whenever you're collecting the monitoring data out on the reef, or this can be when you collect corals from your tank experiments, or from the nursery, from the wild. When did you sample these bad boys? You pretty much always need to do this. I've never seen anyone not, so we're gonna put it in S tier. And I can already see this can get easily out of hand. We have the first 3 and we're already in S and A tier. I don't want everything to end up in S and A tier. Considering most of these metrics I've collected for my studies, I'm probably gonna think they're a little more important than they might be. But as we add more, I'm hoping we can like bring some down. Let's look at processing date next. So this one refers to what I had in mind for this variable. This is when you have all your samples. So this is after you collect your samples and you start processing them. So for DNA studies, for instance, if you're collecting DNA samples, this would be like when you do your extractions or any of your physiological assays, anything in the lab. I kept track of this because, again, batch effects. I want to keep track of batch effects. Thankfully, I didn't see any. I would want to put this in A tier, but for now, I'm going to put it in the B tier. Again, I might change my mind on that one. Oh, Lauren, hello. Agree, sampling date is super important. Yeah, I've never known anyone to not do that. I feel like it would be illogical. It would almost, I don't know, it doesn't make sense not to. Okay. Next up, we have GPS coordinates. Huge deal if you're doing field work. Enormous deal if you're doing field work. So this is like where you actually collect your samples. So similar to site location. This one's kind of tough. I wouldn't say it's always necessary, but you're pretty much going to be doing it, especially for field work. You're pretty much going to be collecting GPS data. But I'm not going to say it's critical, because if you have, as long as you're at least keeping track of site location, like non-specific, like a reef name like Chica Rocks or Pickles Reef, that can be fine. I think it's a little more important if you're trying to show differences on like a finer scale. So like within reef, or yeah, if you're really trying to tie geography, latitude and longitude, climate zones basically, then I would want it. But precise GPS coordinates, it'd be a nice to have. Which I said was like C tier, I think, when we were starting. So we'll move it to C tier for now. You can, I'm always gonna say we keep revisiting this. We should. We'll see. Next up, photos. This is one I had in mind from the start. From this like conception of doing this live stream, I was thinking of photos. This could be site photos. This could be sample photos. This could be photos of you and your team doing stuff out in the field, doing stuff in the lab. Any photos, instant S tier. Instant S tier. You should be taking photos of like everything. I wish I had taken more photos for a lot of my projects. And I had lab work and I had field work. I wish I had taken more photos for both of them. Because sometimes when you're going through the data, things don't always make sense. And I'll have a story for that in a sec. Things don't always make sense. And I wish I had a photo record of things to tie back to help explain it. Okay, maybe I have two stories. So one lab story, one field story. The lab story is one of my chapters. Some of the data we were getting, we were looking for microbial communities that were indicative of coral bleaching resilience. So did certain coral colonies that had certain microbial communities, were they more likely to survive a bleaching event or more likely to die? The ones that survived, our data right now show that microbes associated with the endolithic algae in the skeletons were more present in our resilient corals. And I remember when we were processing the corals at the time, we were removing the tissue, there were colonies where the skeletons were green. Like they had a crazy amount. I use the word amount loosely. They were green. Their endolithic algae was like happy, I guess. And I wish I had kept, like, if I had taken photos of those, then I could tie that into were the corals with those green skeletons, were they also the ones that had like greater resilience and amplicon sequence variants for the endolithic algae, Ostriobium. Now I did keep track of it in my lab notebook, but not as consistently as I probably should have. We had many people airbrushing the tissue, and I'm not blaming anyone for this. And it was hard to keep track of that because there were a lot of corals to go through. So that's the lab story. I wish I had taken photos of that. Also, just photos of people doing work. I wish I had more photos of me and my team doing work. I have no photos of like myself in the lab. Otherwise, I'd be posting them all the time on our social media because like that would be really nice. But I never did that. So that's a real bummer. So then we have the field-based story. Again, I didn't take enough photos of us doing stuff in the field. And I wish I had. They're like fun photos of the reef, like gorgeous photos of the reef, but not a ton of people doing actual work. I have some when I was field sampling in Roatan, because I was also doing outreach for that project. And so I needed photos of everyone doing the work. But again, they're like no photos of me. And for that project, I was tying in anthropogenic sources of stress on the reef, mainly like land usage, so coastal development, fishing pressure, sources of like localized pollution. And I wish I had taken more photos of the site, like as we're preparing to go on the dive, just like a quick, almost like panorama of the coastline or of other things, just to help remind myself as I'm processing the data later, like, oh, what did this site look like again? Like I have all of the health metrics, like coastal development, was it like really bad? Were there sources of pollution like marine debris? I didn't do a consistent job of that either, and I wish I had. So that's why I put photos at the top. That's an S tier for me. Regrets. I have regrets. Okay, this next one, PAM, pulse amplitude modulation, FE over FM, whatever you want to call it here. This one's tough. I know I said I was avoiding physiology, but I consider PAM a little different. A little different, especially if you're doing coral host. If you're doing a symbiont study, symbiont stress, like you really should be using PAM. If you're doing a coral stress study or any like thing where you're any study you're kind of manipulating the hosts or the holobiont, I would almost say you should do this. Just to see, like, to keep track of what else could be a source of variation in your data set. So I'm going to put this as C tier for now. It'd be nice to have. It would be good to have. Again, this one is a little more context dependent. But for one of my chapter with all of the cervicornis fragments, the microbe one I was mentioning before, we kept track of PAM, and it told a bizarre story. We were trying to use it as a primary physiological metric, and it ended up giving us really weird information. But it did uncover something interesting in their study. So we had these corals, in tanks, we ramped up the temperature of the water to elicit a bleaching response, and then let the water cool down and let them recover. We took PAM from the moment we put them in the tanks to the moment we harvested all of them. And the PAM actually uncovered that our corals were still stressed during their acclimation period when they were getting used to the tank conditions. And that ended up being really important because we got really mixed messages across all of our physiological data. And because we had that PAM data from the start, it kind of helped paint the picture of like, oh, our stress response was weird because the corals were already stressed before we even increased the temperature of the water. So it'd be good to have. If you're doing some kind of host experiment, it'd be good to have. Don't necessarily need it all the time. Probably a good idea. All right, back to the field. Reef rugosity, oops. So this is like the change from like the maximum height of the reef structure to like the nooks and crannies in between reefs. So how high and how low. So how shallow to how deep, like that range. I did some work in the dry tortugas. We were doing DRMs, and so we were keeping track of rugosity. And there are plenty of studies showing that rugosity is a decent to a good sign of reef health, because it's showing you basically how the corals are accreting onto the reef, creating structures, that three-dimensional habitat. I wish I had collected this when I was doing my field work. We were just collecting sediment samples, but we were running transects to also do benthic cover. And especially with the reefs in Roatan, they have some serious rugosity to them. And the way I laid my transect out was just linear. I didn't account for, I chose this. I didn't account for rugosity. Like the transect line was completely straight, even if it was crossing in between two coral fingers. Kind of wish I had collected it, or again, at least had like photos or something. I'm gonna put this in D tier though. I was able to get away without it. Obviously, if that's what you're going, if you're studying rugosity, this could be a direct variable that you want to measure. In that case, collect it. But I don't know. Probably don't always need it. I've done other field work, you don't always need to use it. All right, we are, okay. Making progress here, we're making progress. All right, let's do site characteristics next. I talked a little bit about this already, that I was keeping track of site characteristics in the field. So keeping track of surrounding land use, coastal development, marine debris, fishing pressure, a lot of anthropogenic sources, but there might also be other things like eutrophication, which could be anthropogenic, but also can be naturally occurring as well. So the study I had used this for, we were looking to see if microbial communities could indicate whether a reef that was receiving coral outplants was actually recovering over time. We didn't end up getting the time series, we did not end up getting the time series data, which was unfortunate. But I was also keeping track of site characteristics almost as like baseline to like, okay, how healthy was this site when we did collect it? Was it a healthy reef? Was it a degraded reef, which included the benthic composition? And then we could track it over time, like, okay, this degraded reef is shifting to a reef that we had designated from these site characteristics as healthy. It's moving, the communities are moving from that degraded state to a site with better traits, better coral cover, no marine debris, no fishing pressure, no coastal development. So that in a way was in a way that one was a direct measurement, so a little context dependent. But again, something that we'll make this the first F tier. It's context dependent. You can probably get away without it. Or like I wouldn't bother with it. That's what F tier was supposed to be. It's like, I'm not gonna bother with it. Unless I'm actually doing it. Eli, a tier to collect, but maybe not as important to include in papers. That's a good way of thinking about it too. Because I don't know if I'm actually writing up the site characteristics. I haven't read the paper in a while, the manuscript I have. Ooh. I like that, Eli. Because I had a figure for it. I ended up cutting it from the paper. I don't even know how much I like. I have the table that describes the site characteristics, but it's brief. Yeah. All right, we're sticking with the field for now. Why did I include fish abundance? That's a direct measurement, folks. That's a direct measurement. I don't want to focus on that. We're gonna exit. You can complain if you want. I'm sorry if you collect fish abundance surveys, but we're trying to think of everything not a direct measurement. Like this one is good. Personnel. Who did the collecting? Who did the sample processing? So who was out in the field doing the surveys, collecting samples? Who was the one doing the DNA extractions or the PCRs? This should be accounted for. But it's definitely A or S or A tier. I was very picky about this. I made sure to always collect this information. And it's a good one for batch effects. I'm gonna put it in S tier for now. If you guys think it's less important, I'm willing to listen. I'm willing to negotiate on this one, but I'm gonna put it S tier for now. Funny enough, this is great. I had a lot of my undergrads actually would get better results than I would on certain lab methods. It's kind of hilarious. Lauren, you think it would be A, not S tier? I can see that. I can see that. Because unless if it's that sensitive, you want to keep track of it. But again, okay, so it's not the main story. It's not going to help you completely out. Okay, I'll go with that. We'll switch it to A tier. We'll switch it to A tier. I'm with you on that one. Do you guys keep track of who's doing it? Do you have that in your spreadsheets? I always did. It's easy to collect. There's no reason not to. Okay, so this one, it's easy to collect. There's no reason not to, but it shouldn't make or break the story. That would be A tier. Yeah. All right. Cool. All right, let's go back to, it's easy, but I don't think it's as important as most of the other data. I'm not. Lauren, do you mean it's not as important as your primary data, your variables of interest, or comparing it to other metadata? Because we're trying to compare them to each other on here. Not so much in comparison to your variables of interest. I'm comparing it to what we already have in the chart. Okay. Then we'll keep it in A. We'll keep it in A. Awesome. Ooh, okay, then we're going to go to one that's related. We're going to jump ahead a little bit to sampled batch. So this one would be related to the personnel or the person doing the collecting. It's also which batch did it get into? This is probably more important for sequencing data than anything. It's which sequencing run did it get put on? But usually it overlaps with one of these things. So I'm gonna put it in B tier. I'm gonna put it below who's doing it. Although if it's the sequencing run, that's if you're doing a sequencing run, that's your biggest variation that you're gonna have. It's probably between sequencing runs. Done hundreds of extractions, but it's usually the sequencing run where it gets messy. Leave it there for now. We'll leave it there for now. Let's go back to sample site. So out in the field, where are you collecting your samples from? We've already done GPS coordinates. I think sample site is more important than GPS coordinates. Unless you're doing fine scale or really like a biogeographical comparison. Excuse me. I'm between A and S tier for this one. I'm thinking S tier though. I think S tier for this one. It encompasses so many things. And you usually find something interesting in between sampling locations. Oh, and you both agree. Okay. Both of you say S tier, S tier, S tier. Cool. Glad we're all in agreement there. Those usually uncover the really fun stories too. Like, they're usually important stories, but like those are usually the ones where you plug it into R and you're like, what are we gonna find? And it's usually something cool. So yeah, we'll keep that S tier. If anything, just because it's fun. Sampling time. So when did you actually do that? Collect your samples. My gut is saying D tier on this one. But you can look at it in a couple different contexts. In the field, it could be important. Actually, it's for both lab and the field. It might be important to know sample time because of light levels. In the field, it's usually it could also encompass tidal cycles. But sometimes it's more of a headache than anything. I've tried to like be diligent with keeping track of sampling time. It's a headache. I think it depends on your question. Of course it does, Lauren. Of course, it always depends on your question. If you're doing like a diurnal cycle or a DL cycle of your study, your time is going to be super, it's going to be critical. But that's going to be like your main question. If you're collecting this data on the side, I just don't know if I would. Again, if I were to do my field study again, I probably wouldn't bother. Although for that one, I did take photos of the time when I collected them. That's an easier way to do it. That's a pretty easy way to do it. But like I said, I didn't really analyze time yet. So, All right, next up we have benthic cover. So this is coral cover on a reef. This is macro, algae cover, whatever, all of it. Anything that's on the bottom of the ocean. And this could be a direct measurement. Like I had fish abundance above there before. That was way too direct. I don't want to delete benthic cover though, because I think it could be a useful accessory measurement for certain field studies. It's almost like site characteristics. I'm thinking it's a little too context dependent for my taste for what we're doing here. But I like knowing the benthic cover too. I'm gonna put it in C for now, because it's kind of a fun thing to measure. This can be really used for most marine studies. If you're not doing a coral community study, then C. Okay, yeah. C or lower, so C would be your highest. Yeah, benthic cover is really useful for a lot of different marine studies. I think I'll leave it here and see. It's fun to measure it too. I enjoy measuring benthic cover. All right, YouTube will appreciate this next one, I think. We'll have a fun time with this one. Molecular quantification. This could be the qubit, this could be the nanodrop, quantifying whatever you're measuring. Whatever you're measuring. Oh, hang on, Lauren. I mean, the whole thing, like this metadata must-haves, can be used for most marine studies. Yeah, true. Yeah. And perfectly happy if people use it in other systems and comment on it. That'd be great. Molecular quantification. Eli says S tier for molecular quantification. Really? Really? You say S tier? Lauren says B. That's funny because I was thinking A. Let's hear the arguments. So what's going on in my head? Why it's not S tier? It's an important measurement because for most things you need to know the level of quality of your molecules before you do anything with it. Like your DNA or RNA has to be of a certain quality to sequence it well or quantify it, whatever you're doing. So you're probably going to check it. And Eli, in your case, I can really understand why, but Lauren, your answer surprised me a little bit because you did RNA work. You have to have quality. Yeah, Eli, this is assuming you do have molecular data. Yep. So you must quantify it. But do you? So let's think of it this way. Like, let's break this down a little bit more then. So there's like quantification before you put it on the analyzer, like a sequencer or what other fancy, like the end quantification. So let's think of it before that. So like right after a DNA extraction or a PCR, like how do you feel about quantifying it there? Or do you think it's as important? Lauren, if you're using PCR, it's not that important. I agree. I agree with you there. I was very strict on it early on in my PhD because I wanted to make sure my extractions were going well. But especially for my sediment samples, they just don't quantify well. Like it always looks like garbage, but then you run a gel and it's like perfectly fine. It's like totally okay. For RNA work, it's definitely really important. I'm sure, and that's why I avoided RNA work as much as I could. Or genomics work, I guess it's important too. If you need like high molecular weight DNA, probably. If PCR is not working, then checking your DNA extraction quality is the first thing you have to do. Yeah, you're probably right. So might as well do it first. And I did it a lot of the time. But I found with bacteria, you could just do the gel. If it's on the gel, you've got it. Nanodrop sucks, so maybe if you only use the qubit. And I think that is going to be my final opinion of it. Is Nanodrop does suck. Like, it's so inaccurate. If you're referring to the qubit, it is important. Yeah, you have to use the qubit before you use a sequencer or anything. So all in all, I think I'm gonna leave it in A tier. It's the responsible thing to do, and before the end quantification, you're probably gonna have to do it in some level. So you're gonna be doing it. But in terms of like early stage, you might be able to get away without it. Might be able to get away without it. Okay. That was a good discussion. I liked that one. That was fun. I knew you guys would enjoy it. I knew you guys would enjoy that one. Got water quality next. So this is turbidity, this is alkalinity, this is salinity, nitrates, ammonia, anything you're keeping track of. I had originally thought of this more in the context of a field study when I added this in, because a lot of the water quality stuff I did was for field work, but it can easily be done in labs too. This could also be one of those, it could be a direct measurement. Like, this could be your interest, like, how does turbidity affect the FE over FM? I think there's actually, there should be a paper coming out on that soon, I think. But I'm thinking water quality is a B tier. It's usually helpful. You don't need it. But for, this is coming from an ecologist here, it's cool to know. Ooh, but is it more, okay, I'm getting ahead of myself. Is it more important than benthic cover? Would I choose water quality over benthic cover? Or PAM, or GPS, or any of these others? I'm gonna move it to C tier. I keep it in line. I feel like it's very similar to benthic cover. Extraction noobs hate to see low concentrations on the nanodrop. I mean, don't we all? We would love to have that nice sweet spot, that sweet bell curve. Not bell curve. That sweet DNA curve. I wish all my samples would show some concentrations. My sediment samples would always be like nothing, but there was DNA in them. Yes, water quality is more important than benthic cover. I'm guessing you're thinking in terms of tanks. And I like where you're thinking. We'll put it back in B. Just for the tank experiment, folks. Just for y'all. Temperature. Our coral. Agree with Eli for coral studies. For coral studies, sure. Now we've got temperature. Like, this is why we do coral research, is because of this, is because of temperature. So If you're doing a temperature stress study, duh, you're going to keep track of this. Super important. For others, for field studies. Did I keep track of it for my? You usually do. I'm thinking S tier on this bad boy. I think you just should. I think for most coral research, you're probably going to be keeping track of temperature for some reason. Not even if it's a thermal stress experiment, you're probably going to keep track of it. I didn't necessarily need it for my sediment microbes, my reef microbes. I have the data. I have it. It could certainly play a role in that too. It's so easy, why not for everything? Yeah, absolutely. It's such an easy thing to keep track of. If you're gonna have a dive computer on you, probably has a temperature gauge. It's there. You have tanks, they're gonna have temperature gauges in them. You have incubators, you set that temperature. Yeah, we'll make it S tier simply for how, I mean, it's relevant for nearly everything, and yes, it's so easy to keep track of. Absolutely. Disease and mortality. So if your corals, your anemones die, they're diseased. I should, you can include bleaching on this too. I don't know why I didn't. So any kind of like unhealthy phenotype, essentially. It's funny, for one of my projects, we hadn't considered mortality as like a phenotype because we were keeping track of physiological data. But because all of the phys data was so wonky, we ended up using mortality as a phenotype. Can we put another S tier? Lauren agrees, S tier. I feel like we're putting so much in the S tier. But it's and this is, let's put in the context of temperature again. Like, it's pretty easy to keep track of. Did they die? A lot of the times, if you have something that dies, you have to pull it from the tank anyway. So you're going to keep track of that. But guys, we can't have everything in S tier. We're going to have to move some of these, I think. Oh, maybe we'll do that as we finish up here. So color, this is like if your corals are paling or if they're bleaching, score to keep track of. I rate this very similar to Pam, but it's easier to keep track of than Pam, and you get photos. You're going to be collecting photos as you do it. But you might not always. I'm going to put in B tier for now. I might want that in A. We'll revisit as we compare the others to each other. All right. Diversity. So this is like species diversity. Oh, there's a lot of very important metadata that is needed in a system that is so complex. Yeah, why did I decide to do this? Why did I decide to do this? They're all, I mean, they're all important. They're all important. But if I had to go out and perform a study tomorrow with no information, I guess these are probably the things I'm going to keep looking for coral research. Species diversity. So this can be corals out on the reef. This can be overall species diversity. This could be microbial diversity. This could be anything. B for color or less. Okay, cool. We're chilling then. I feel like diversity should be low. Because it's a headache and it's not always helpful. Especially with like microbial diversity, it's so nuanced. It can be, you do have to calculate it, but it's kind of annoying. I'm gonna put it C tier. Mainly because I hate keeping track of it. Diversity in C, super time. Okay, so you agree, super time consuming. It's, yeah, I think that one, like it is important. In a lot of studies, you have to include it. It's just a headache. I don't like doing it. I don't like doing it. C is like if you have to. If you have to collect it, do it. You don't, maybe try and get away. Lastly, depth. So depth of your corals, depth of your samples, like I was collecting sediment samples, what depth did you collect in that? This, I'm between A and B on. I'm in between A and B on this one. I don't think it's ultra critical. It's certainly not like S tier, I don't think, but it does have implications for light levels, for physiology of, I'm thinking of a lot of different reef things here. Like for microbial communities, depth does matter. Depth has to be in the same tier as temperature. Shoot, you're right. You're right. You're right. They're usually linked. Especially with bleaching. You're totally right on that, Eli. Okay. Really important for knowing what Symbiodyniaceae might be present too. Yeah. Move photos to B. I need to hear the argument on that. I'm very reluctant to move photos to B, but I am open to ideas. Because what we're gonna do next is now we're gonna start comparing everything to each other. We've got another hour or so. We can compare everything to each other. Essentially what this should probably look like is kind of bell curve shaped, so we should see more in the B tier. S tier should certainly not have all of them. Should not have the most. So we need to figure something out here. Move photos to B. Oh, you already read that. I say photos would be A, because your project can get away without them. As important as they are for data processing and for presentations, you can publish a paper without photos of sites. Yep, you're right. You're right. As sad, because that was my biggest regret. Okay, Lauren, I'll move photos down. We'll put it in B. We'll move it to B. Twist my arm. You did. You had to twist my arm in that one, but I get it. But honestly, okay. There's still a lot in S tier, but I don't know if I would move any of these others. Important, because depth is an important consideration. Disease and mortality is usually a great phenotype. Temperature, like the central part of our research, so you should be collecting it, and it's super easy. Depth is super easy. Disease and mortality are super easy. You'll probably take photos of those, but sample site, that's really how you're going to name your data sheet. And there's so many variables with it, so that has to stay. When you do it, yep, if there was one like metadata, like data about the data, sampling date is going to be the ultimate one. Because it's like, when did you do the thing? It's basically a row. Your rows are essentially determined by your sampling date. Either that or like individual, of course, the sample itself. The sample itself is a row, but a lot of it's going to be sample date. I think all rest of S need to stay. Okay. Sampling date to A, since not all studies are time or seasonally dependent. I get what you're saying. I get what you're saying. But almost everything can be dependent on season, sure. I think without sampling date, you lose just a lot of context. And you can have batch effects. I think we're going to leave sampling date. We're going to leave it. We're going to leave it there. So let's move on to our A tier. Is there anything we want to move? So genotype, this could be host or symbiont. If you're doing a coral-based study, I think you have to have this. Because this deals with replication. I think you need to know the genotype. It has important considerations. It's an important source of variation. If you're worried about like difficulty of actually knowing a genotype, some of it can be a headache. Coral host, if you're collecting stuff from the field, you can usually do it by distance. If they're already in nurseries, you probably have already assigned a genotype to them. If genotype is part of your question, you're probably going to use molecular data, SNP chip or something, to actually genotype them. Cultures, symbionts, you probably have a better... You two have a better idea of the importance of symbiont genotype, like keeping that organized. I'm sure it's critical. But there might be certain cases where you don't need it. Which is why it's not in S tier. I feel like personnel could be B if we are comparing it to the other categories in A. Yeah. I can get on board with that. Yeah, I can get on board with that. Because while a person like could make or break, like there's bias. There are other ways you might be able to figure that out, or at least account for it. I'm okay with that. tank, I do consider very similar to genotype. If you have different tanks, things can happen. Bad stuff can happen. Whether you know it or not, little things can add up. Molecular quantification. Should we revisit this one? Could this go to B? We're gonna have like nothing in A. We're gonna have to move some stuff to A or something. I don't know. But at least we have more than S now. S is not the hugest category. Eli, are you gonna die on this hill? Molecular quantification to S. But do you report it? Lauren says keep it in A. You might report it in your supplemental depending on what you're up to. I think we're gonna keep it in A. I don't think it belongs in S. Oh my god, Lauren, we convinced him. We convinced him. It's the perfectionist in you, Eli. That's what's getting you here. All right. B tier, anything we want to move. Got processing date, which is very linked with personnel. So essentially those are the same, so those could stay. Sample batch, these are all basic. Okay, so basically B tier is like batch effects. We could literally just probably call B tier batch effects. Water quality could cause batch effects. And if you're not like direct, it's not a direct measurement. Color would not be a batch effect. But like basically these are all batch effects. Besides color and photos. Water quality could be related to tank though. Essentially that like changes in water quality would be a tank effect, would lead to a tank effect. Color could be moved down. Oh, you're right, because Pam, I related it to Pam basically. Cool. That's good. Photos, do I want... Oh my god, I'm going through a full like reverse. Do I want to move photos to C tier? Is that bad? The one thing I wanted to talk about with this was photos. I put it in S tier and now I'm like, oh, we moved it to B. Murph agrees that sampling date should go to A. Well, I can't really argue with Murphy, can I? Do you ever not take that data though? Like everything I've ever done, date is in the sheet. It's in the data sheet. Murph is lying. I think Murph is lying. Sampling data is almost like beyond. Like it almost shouldn't even be a part of this because it's just so intrinsically linked with data collection. Like, I can't imagine a scenario where you wouldn't do that. No, that's true. Date should stay S. Oh, we'll give my best to Murphy either way. He put a good argument. Wait, Murphy's a she, isn't she? I'm blanking. Imagine not writing the date down. Exactly. Like, my brain cannot comprehend why you wouldn't take the date. I think it's perfect now. Just B tier, or you think the whole thing? We think we're done here? What else had we here? Sample batch, water quality, photos. We already moved personnel. GPS, PAM, color, benthic cover, and diversity. Those were probably a little too specific, but I just like them. Well, I don't like collecting diversity data. I know that's irresponsible to say. Rugosity, sampling time. I feel like rugosity and benthic, I don't know why I included rugosity. I include that just because I collected it before. Interesting. Yeah, I don't know if I would move anything else. Are there any other metrics you guys have that I may have missed? That are not like super context dependent? Is there other stuff like you guys collect alongside your data that I just didn't think about? It's very possible. There's a lot of things to measure. I think it looks good, the whole thing. Cool. I think you covered most of them. Yeah, this was, okay, cool. All right, so we have our list. So here's our finalized list of coral research metadata things you should always be keeping track of. Should be like a given on your data sheets: sampling date, sample site, temperature, disease, mortality or bleaching, and depth. Always good to have. Number of hydroid stings. That is important to keep track of, although you may lose count. That's a lot. A should be, and like usually S tier things are like super easy to collect, so that's why we included them as well. A tier is things that are pretty critical to your study, can be kind of a headache to keep track of or quantify. But you'll use it for most studies. Maybe not some field-based ones, but for most things you're going to be keeping track of. And they could be useful for models and certain batch effects. Although we ranked tank higher than other potential batch effects. So our B tier is like, you should probably be collecting these, but it won't like make or break your analysis. Like responsible to keep track of, but you won't die. C tier, GPS, PAM, FEO over FM, color, benthic cover, diversity would be nice. Probably not going to be the first thing you're analyzing, whatever your question may be, unless it's like directly measuring benthic cover and its effect on blah blah blah. D tier is like it's kind of a headache to do. You probably won't use it in your analysis. It's just an extra thing. And then F tier, it's like if I were to do a study again, I wouldn't even include this. And certain, of course, with the whole thing, we get to the phrase that I hate the most. It depends on the question. Of course, with all of these, it's going to depend on the question. But our goal here was essentially, if I were to have to go and do a research project tomorrow in the field or in tanks, here are the things I would want to be keeping track of, regardless of the question. I need to know the sampling date, where I collected things from, temperature, because corals, temperature is everything. The health of the corals, essentially, and depth, pretty similar to sample site. Instead of like flat and long, it's up and down. Important to keep track of. So I will go ahead and I'll finalize this. I don't know, I'll either make a spreadsheet or I'll just do a screenshot of this. And I'll put it in the community forum, I think, to start. I'll also send it out in our email blast. I'll send it out probably next week and provide all the context that I just talked about. So you guys have this. You can print it off, put it on the lab fridge, in case you do have to go out into the field tomorrow on a whim and you don't know what the heck you're doing. This could be useful. That is an important point, Eli, because the more you get, the less data you probably collect. Yeah, if I'm getting stung by hydroids, I'm caring less and less about my data. I just want to get out of the water. Sure, Lauren. Yeah, happy to do this. This was super fun. I'm glad you guys could make it. It would be a lot less exciting if I didn't have the discussion and feedback. Should we do another tier list? Do you guys have any ideas? What should we rank next? Coral species? Should we do coral species? Caribbean species? That could be a fun one. Not today. Not today. Yeah, we could do coral species. We could do lab techniques. Oh, we could do so many different things with this. Coral species tier list. I had a feeling you would like that one, Eli. I think we all know where we'll put the acroporids. I don't know enough about the Pacific species to really rank them. So we'll probably have to do the Caribbean ones, but there's too many of the Pacific too. That would take all day. Alrighty, folks. I think we'll wrap it up here for today. Thanks for coming out. I'll see you guys next week, probably. See you guys next week. I'll post this as soon as I can. And then we'll keep doing this. Don't have as much input on the coral species, but love to learn F. We put F's in chat. All right, guys, I'm signing off. Have a good rest of your Friday. Have a good weekend. Bye-bye.